Hello, everybody. Welcome back. It's me, Yajie. Next talk will be given by Dr. Adi Stern. Adi did her PhD at Tel Aviv University, and after her postdoctoral fellowships at Weizmann Institute of Science, UCSF, and UC Berkeley, she returned to Tel Aviv and founded her own group there. Her lab studies viral evolution and host immune responses with the help of computational biology. In today's talk, she's going to introduce her research about finding the pattern of viral transmission by analyzing the full genome viral sequences. Let's welcome Adi.
Hi. So thanks really, Arji, and thanks for the opportunity to give a talk. I was hoping to visit Germany, but here I am talking by Zoom, and I'm still really excited to present my work. And so I'll be telling you today about how we studied coronavirus, not surprisingly, SARS coronavirus 2, and how we used sequencing to understand how the virus spreads in Israel. So I want to actually begin with a very brief introduction on COVID-19, which is the name of the disease in Israel. And so what you see here is the number of cases across time, and this is, so the first case in Israel was detected somewhere around February. Then the number of cases began to increase exponentially, as occurred in most of the world. Somewhere in March, lockdown was implemented. It took a while for it to work, as you can see, but afterwards, cases really declined very, very dramatically. And Israel was really considered like textbook success for how lockdown can eradicate the virus. As you can see, the virus almost hit zero in May. And unfortunately, the rest of the story is less positive because this is what is happening today. And so cases started to increase in June. And as you can see, there's been a dramatic increase. And it's actually even worse. I couldn't even find a figure to show how much worse it's become. So in the past week, there have been, if you see here, this is 2,000 cases a day, and we're now at 5,000 cases a day. A very, very dramatic decline in the situation of Israel with the coronavirus. And so most of the story I'm going to be telling you is about this part of what went on in Israel, about the first 2 months. And now I'm going to actually be telling you my personal story. At this very moment, I'm in quarantine for 2 weeks because my kids, whose picture you can see here, were exposed to a verified SARS coronavirus 2 patient. Since, of course, they can't enter quarantine on their own, I'm in quarantine together with them. And so far, everyone is feeling good, but we're at home. And so that's part one of my personal situation. Second part of the more global situation is that very unfortunately, as of tomorrow, Israel is entering another lockdown. So we're entering the second lockdown. We're the only country that is entering a second lockdown. This is very, very sad. I think it's very, very sad locally for Israel and globally, as I know that this is a pattern that other countries are also experiencing, the second wave. And I'll try and end this talk by hopefully the positive messages that I do have about how we can control the virus. Okay, and so now after that kind of personal interlude, we'll now get to the scientific part of the talk. The scientific part of the scientific work actually took place during March and April of 2020 during full lockdown of Israel. And so I'm beginning with my thank you slide because I think this is kind of the most important slide that always people don't have time to get to it. So you can see that this was a huge collaboration. It was led by these graduate students, all shown here, Danielle Miller and Omer, who were computational biology students. And this is Moran, Noam, and Talia from my lab getting ready to accept samples from patients. And as you can see, that they're in full protective gear. We collaborated with a huge number of medical centers in Israel and with the Israeli Ministry of Health. And of course, we have to thank the funders for allowing this research to have taken place. Okay, so that's my thank you slide. And now I want to give you some background about corona, about what people sort of call the coronavirus. And so as I hope most of you know, the disease is called COVID-19, coronavirus associated disease 19. 19 is from the year 2019 when it was discovered in December 2019. The virus is called SARS coronavirus 2. This virus is part of the coronavirus family of viruses, which is well known in the field of virology. There are 4 strains of coronavirus that cause typical common cold. What we all experience is not the flu, okay, but it's kind of just this typical bad feeling that most of us feel in the winter. You have a runny nose, you feel a little bit bad, and usually most of us overcome it within a few days. There are 2 other members of the coronavirus family, which are SARS-1 and MERS. And so these are another 2 viruses that also jumped into humans. They also caused more severe disease, but they caused a much more limited outbreaks. And I can elaborate later if anyone is interested why there was a difference between this virus and these other 2 viruses. But that's kind of another sidetrack to this talk. And some more facts about about coronavirus, about SARS coronavirus 2. So you can see the level of genome identity between different viruses. Okay, so between SARS coronavirus 2 and the common cold, there's about 50% identity, so it's relatively low. Again, it's still strong homology, but relatively low. SARS coronavirus 1, there's already 80% homology. And bat coronavirus is a specific strain of bat coronavirus, harbors 96% identity to SARS coronavirus 2, and that is why it is thought that the most likely explanation for how this virus entered the human population is by contact with bats or through some intermediate population. There's some discussion about pangolins possibly being the intermediate population. So the virus might have jumped from bat to pangolins and from pangolins to humans or some other animal that has not been discovered. Another important measure is the evolutionary rate of this virus, which is 10 to the minus 3 nucleotides per year. So this is the number of changes in the virus genome per year. Again, if you multiply this by the genome length, you get the actual number that you get per year. This is a relatively low number, as I'll soon show you. But before that, I'll just, again, a brief discussion about how this virus looks. And so this is the structure of the virus. The most famous protein in the virus is the spike protein, which is this external sort of spike. That's actually how this virus got its name. Corona means spike. And so the spike protein is the external part of the virus. It's what antibodies recognize. It's what our bodies recognize. And it's also what allows the virus to enter our cells. So there's a receptor domain here which binds the receptor, which is called ACE2. This ACE2 is expressed in our lungs and in different respiratory tract cells, and this is how the virus enters. And so there's been a lot of focus on this particular protein because it's really so important for the biology of the virus and for the immune response, which is very critical, especially for those who are now designing vaccines. Okay, and so there are different various interesting facts about this virus, and any of you who are interested are welcome to enter this eLife paper that came out on numbers describing the coronavirus. But I'd just like to go back to what I discussed about the evolutionary rate, and so this is some comparison of evolutionary rates of different RNA viruses. And so as I mentioned before, SARS coronavirus 2 has an evolution rate of 10 to the minus 3 per nucleotide per year. And if you multiply that by the genome length of the virus, what you get is 1 mutation per 1,000 bases per year. On the other hand, influenza virus has 2 mutations per 1,000 bases per year. So you can see it's double, its rate is double that of SARS coronavirus 2. And HIV has actually times 4 the amount of mutations. So 4 mutations per 1,000 bases per year. So that means that relative to most RNA viruses, SARS coronavirus 2 is a relatively slow evolving virus. This might be good news in the context of mutations to the spike protein and to the stability of this protein. Although again, I have lots of kind of additional things to say about that. On the other hand, I can say that from the point of view of my work, This made my work harder because, as I will show you, I used the mutations in the virus as kind of markers which allow us to track how the virus spreads. And having said all that, still the rate of evolution of SARS coronavirus 2 is still much, much, much higher than humans or any other organism out there. Okay, so it's still a very, very high rate of evolution. Mutations. So this paper came out on the 18th of February, and I think it's a very, very important paper. And it says, we shouldn't worry when a virus mutates during disease outbreaks. And this came because there was this kind of, and there still is, every once in a while the media comes out with a statement, the virus is mutating, there are mutations on the virus, and then everyone gets really, really scared. Because as described here, the word naturally conjures fears of unexpected and freakish changes, right, this word mutation. But actually mutations are a very natural part of the life cycle of a virus, right? So all viruses mutate, and they mutate all the time. And the vast majority of mutations that we observe are neutral. There's no change to the function of virus. This is probably true for every single organism out there, but specifically for viruses, it's actually been observed and shown that really the vast majority of mutations are neutral. And so this is an important thing that I'd like you to keep in mind as I continue on this talk. Okay, so let's continue and describe the motivation of the study. And so I'll actually say that our original motivation was something that we didn't completely succeed in obtaining, as you'll see towards the end of the talk. But the general motivation was to gain a better understanding of the epidemiology of virus spread. Okay, and so there's this newly emerging field which is called genomic epidemiology, which is using genomes of viruses to track how the virus spreads in the population. And so there's a lot of information in the genome of the virus in the form of these neutral mutations that I just described, and I'll elaborate on in a moment. And this information really allows us to track viruses. And so our original question was actually about incidence. And incidence is the total number of people that are infected in a population. And so originally there was this huge debate and huge discussion around how many asymptomatics are there. And so we knew that when we will follow, we were generating information about how many people are infected. Most of the people that we detect are those that have symptoms, those that are hospitalized, and so there's this kind of bias towards the symptomatic patients. And so what we wanted originally to know was how many asymptomatics are there and what is the total number of people that are infected in Israel. And this, of course, has a lot of implications for policy making, right? The total number of people will tell us what the mortality rate is, for example. So the mortality rate is basically the number of people who die from a disease divided by how many people in general are infected. And so that was actually our original motivation. Our second motivation was to estimate the basic reproduction number, which I'll be elaborating on later, but I'll again say that the basic reproduction number tells us how many people every infected person goes on to infect. And so again, this is a very, very important number for understanding how the virus spreads. And so the way epidemiology traditionally works is something which is called questionnaire-based. And so this is something which is very, very slow and may often be biased. Okay, and so if you're sick, you get a telephone call from a nurse, and the nurse will start questioning you who you met and how you met and when you met. And eventually the idea behind this questioning is to understand who infected you and how many additional people you might have infected. And then sort of contact networks may be generated from these questionnaires. This is a process which is very, very slow, and additionally it might be biased because unfortunately people don't like telling, they don't necessarily like telling the truth about who they met and why they met. There's a lot of research into the problems that there are in this questionnaire-based mode of study. And our idea is that a rapid outbreak really requires a rapid, unbiased response, and that sequences are the ideal measure to do this. So sequences don't lie, right? And sequencing can be performed very, very, very quickly, and so that was our motivation. And so what do we know? Again, I kind of gave you some kind of background about SARS coronavirus 2, but here I'd like to show you, again, to emphasize this idea that mutations are neutral. There's something which is called the clock-like evolution of the virus. And so because of the way the virus evolves, because it has a polymerase, it introduces mutations in a sort of Poisson-like fashion. We know that mutations accumulate in 2 mutations per month. So every second transmission, there's going to be a mutation that will fix in the genome of the virus. And this is like the set pace of evolution. And again, of course, this is on average, and there's standard deviation around this and so on. And later I'll show you we have a complex mathematical model that takes all this into account, but this is kind of the intuition behind it. 2 mutations per month. And this is going to be an illustration to describe how these mutations accumulate, okay? And so we're going to be looking at the virus genome and how it spreads, and how the virus spreads across the world. And so we have this presumably the first case, which occurred somewhere in China. We have the genome of the virus. This is our so-called patient 0, okay? And so we actually what I'll show you is that we compare everything. We compare all the mutations to what we call the reference genome, which is the first virus that was sequenced in Wuhan in China. So we have this virus in China, and then this person infects someone else. And again, since every second transmission there's a mutation, in the first transmission we don't expect a mutation to occur. In the second transmission, we do expect a mutation to occur. And then just by chance, this person travels to Europe, and we get this virus that is now spreading in Europe. Again, every second transmission, we see another mutation. Then, just by chance, this person travels to the United States. By now, we have 2 mutations on the viral genome. And then in the second transmission from there, an additional mutation will accumulate. And by now, we have 3 mutations in the virus genome. And the idea, the reason that I'm showing you this kind of toy illustration is that eventually what we do is we use these mutations as barcodes. Okay, so if we see a sequence in Israel that has these 3 mutations, we will assume that the most likely explanation is that this genome originated in the United States. Whereas if we see 0 mutations in a sequence in Israel, we will assume that this genome was derived from China. It's from a patient that traveled from China to Israel. And of course, we try and make these estimates based on as many mutations as possible. So I can tell you that by now, when we sequence genomes, we see something like 20 mutations that separate the sequences in Israel from the sequence in China. When we performed the study back in April, we saw on average about 10 mutations separating genomes in Israel and genomes from Wuhan in China. Okay, and so the first stage of the study is that we collected 212 samples from SARS coronavirus 2. We actually collected more than 212 samples, but some of them were filtered later on. This is leftover RNA from what are called nasopharynx swabs. So these are the swabs that are used to test for the presence of virus. Some of you, I think this has become a kind of famous procedure by now. So this is the swabs are taken from very interior part of your nose. And then RNA extraction is performed to perform a real-time PCR test for presence of virus. And so we use the leftover RNA. Luckily, there's a lot of leftover RNA. And the criteria we employed are 3 different criteria, which are very, very important. The first is that we focused on different dates spanning March to April. No relatives, so we excluded any relatives, any people that might have been father, mother, anything like that, we excluded. We collected from 6 hospitals spanning the entire geography of Israel. Israel is a tiny country, and so 6 hospitals were enough to span the entire geography of Israel. And the idea behind these criteria is that we wanted to obtain a random representative sample of all infected individuals in Israel. Then we performed full genome sequence of the virus from these 212 samples by next generation sequencing. The genome is almost 30,000 bases long. It's actually a pretty long RNA virus. And we managed to successfully sequence all the 212 samples. Again, as I mentioned before, some were not, a very small number of sequences were not full genomes, and we excluded them from the study. And so what did we learn from these sequences? So first of all, I'd like to show you a map of the mutations. And so every dot here, so the x-axis is the position in the genome. The y-axis is the count of samples with a given mutation. And so you can see this mutation that I'm inserting now is somewhere around position 2,500. And it was present in a few, maybe a dozen sequences, a few dozen sequences in Israel. And so you can see that there's this kind of spread. The ones in red are non-synonymous. So these are mutations that affect the protein, that change the amino acid. The ones in green are synonymous. We have a few non-coding sequences, so ones that don't affect the coding region. And you can generally see that this is it looks like a very random pattern, like as if we threw these dots onto the figure here. And this is actually an indication that there's no selection operating here. There's no tendency to some preference for some region where there's more mutations. I will mention one mutation, which is this mutation up here. This is called D614G. It's the only mutation so far which has been implicated in affecting, to some extent, the biology of the virus. And so it's thought that this mutation leads to a virus that is not more pathogenic, but is more transmissible. Again, I can elaborate later if I have time. It's present in 90% of the Israeli sequences, and this is actually true for the entire world. So the strain that is dominant in the entire world is a strain that includes this D614G mutation at the spike protein. And it's thought that this is probably one of the reasons why this virus spread so quickly, because it's a hypothesis at present that this mutation allowed the virus to spread very, very quickly. Again, it doesn't cause more disease. It just presumably is transmitted more efficiently. Okay, it doesn't affect any of our calculations because it's one mutation out of many, many other, and it's basically almost entirely present in Israel. Okay, another kind of intriguing fact that we notice is that 8 sequences in our study harbor deletions, and so these are short regions that were just completely wiped out of the genome of the virus. We were very surprised by this. 3 of the 5 deletions are in frame. 2 of them are not in frame and affect ORF7A and ORF8. So these are 2 deletions that one of them has a dramatic effect. ORF7A is basically wiped out of the genome of the virus. It's very surprising, but we're not the only ones to have reported them. I'm going to skip the sentence and just say that one hypothesis is that ORF7A is a gene that might be necessary only for replication in bats, not for replication in humans. And so what we think this means is that SARS coronavirus 2 might be prone to deletions. Now I want to go on to the next part of our research. What we did here is we compared Israeli diversity and global diversity. And so you see here a kind of a tree representing different samples across the world. Purple is China, green is Europe, and red is the United States. The blue dots are Israel. And I think what you can actually see pretty visually here quite clearly is that most of the blue dots in Israel are clustered inside the United States clades. This was very surprising. And when we see this visually on the map, this is our inferences when we put them on the map, we found that 70% of transmission chains in Israel are from the United States. The rest are from Europe and almost none from Southeast Asia. And we were originally very, very surprised by this, and we thought that this might have to do with travel, incoming travel patterns from the United States, but this turned out not to be true. So the Ministry of Health actually has reports on how many infected returning travelers there are, and only 27% of infected returning travelers are from North America, which is the United States, whereas our data infers 70% of what we called clade importations coming from the United States. And actually what we figured out is what we figured out from this discrepancy is that travelers returning from the United States contributed dramatically to spread in Israel. We do not see this in other countries, so you can see this as inferences were made for other countries. No other country has such a high proportion of red, which is importations from the United States. And what we actually think this means is that this has to do with the way Israel, with the policy in Israel. And so the policy in Israel was that borders were shut down from Europe already at the end of February, but travel was allowed from the United States till mid-March. And so in those 2 weeks between the shutting down of borders with Europe and the shutting down of borders with the US, people returned from the United States. They didn't enter quarantine because they weren't supposed to enter quarantine, and they just traveled around Israel and spread the virus. And we think that this created a huge amount of damage in Israel. And I think my most important message is that if we enter a second wave, you have to close down all borders. You can't have differentiation between one country and another country. The second part of our study was to perform phylogenetic modeling, and this was performed in collaboration with Katja Cole and Michael Martin at Emory University. And what we did here was we fit a mathematical model of what's called the susceptible, exposed, infected, recovered model. It's a mathematical model with various parameters that govern the probability of being susceptible and transitioning to what is called an exposed state, and then to becoming infectious, to recover. And so again, I'm not going to get into the details of the mathematical model, but I'd just like to say that the model takes into account very important parameters of viral spread. The most important parameter is that it allows for transmission heterogeneity, which means that some people might spread the virus more than others. And this is something that we actually know from dynamics of infectious disease, and I'm going to elaborate on this more, so I won't talk about it now. The second thing is that we allow non-infectious periods following exposure. Again, I won't elaborate on this. And the third thing is that we assume that transmission dynamics might change following social distancing. So it made sense that if social distancing was implemented in Israel, we'll see a change in the dynamics. And so first of all, what we did is we estimated R0, which is the basic reproductive number, which I mentioned a moment ago. And so R0 is the mean number of individuals each person infects. And so we infected that before lockdown, this number was about 2.3, somewhere between 2 and 2.5. And that after lockdown, this number dropped to 0.7. And this really illustrates, we see this directly only from sequencing data. The lockdown worked, okay? Just from sequencing data, we see very clearly how the lockdown worked and brought down the reproductive number very, very efficiently. We actually have data that I'm not showing you here, that the number dropped down to somewhere around 0.3, 0.4, which corresponds with this very, very low number of cases that were present in Israel in May. And so transmission heterogeneity, R0 represents an average. And we know that actually there's a Poisson distribution around this average. And even more than that, we know from the literature that there's something which is known as a 20/80 rule. So for many infectious diseases, we know that 20% of infectious individuals are responsible for 80% of cases. And this is in this illustration, okay? 20% of patients are responsible for 80% infections. This rule somehow turns out to be true for a really large number of viruses and bacteria. And it's very, very interesting in itself why this is so, but it turns out that this is so for many, many different viruses. And so initially we implemented this rule. We thought this is probably going to be true for SARS coronavirus 2. And so here comes the question, does the 20/80 rule apply to SARS coronavirus 2? What we have here is the model predictions. We simulated cases under this 20/80 rule. And so this is our model predictions, the orange line. And these are the reported cases, the actual reported cases. And so what you see here is that under our model predictions, if we assume the 20/80 rule is correct, we get less cases than the reported cases. And clearly this doesn't make sense. It doesn't make sense that our model will give less cases than the actual number of reported cases. We expected this line to be higher, either at the same level or higher. And I'm cutting a long story short. We stared at this graph for many, 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 many days. And we tried changing other parameters of the model, and it took us quite a while to realize what is most likely wrong is that the 20/80 rule does not apply to SARS coronavirus 2. And so when we change this assumption, and so we have this parameter called pH, which is the number of high-spread patients responsible for 80% of infections, we want to be able to overlap this dashed line, which is the reported cases, or be higher. We realize that the only way to explain the data is by extreme super spreading dynamics. So between 2 and 10% of patients are responsible for 80% of infections. Okay, so this is actually the most extreme numbers that have been ever reported for any virus. The only viruses or bacteria which is relatively close is SARS coronavirus 1, which has a value which is somewhere between 10 and 15%. And here we actually think the most likely explanation is that somewhere around 5% of patients are responsible for 80% of infections. Again, very, very extreme super spreading dynamics. And I'd actually like to kind of end with kind of perhaps the good news, okay? So actually control of virus spread, this has been shown in 2005 and subsequent patients. Control of virus spread is easier for viruses with super spreading features. And the intuition is that if we prevent large gatherings, we can actually prevent a lot of the virus spread, right? So if we manage to find those super spreaders, and so it's hard to actually find the individuals that are creating super spreaders because we have no idea how to find them. But what we can prevent is from them exerting their potential to super spread. So if we prevent, and that's what I think most countries are doing, right? Preventing gatherings of 10 or more people, we should be able to prevent most cases. We should be able to prevent something like 80% of cases. And this is what this graph shows. The probability of extinction becomes higher the higher the level of super spreading. And so my last summary slide, what have I shown you today? Transmission from US travelers is due to a gap in policies. We find super spreaders. We don't know if they're biological, more virus in certain patients, or social, large gatherings. It's probably a combination of both. By the way, a corollary is that most individuals do not contribute to spread at all. Of course, we have no idea who these individuals are, and so still we have to exert utmost caution and social distancing. And I showed you the sequencing data alone shows the effectiveness of shelter-and-home measures, and were implemented in March 19. They are going to be implemented as of tomorrow again in Israel, and I'm crossing fingers that we, together with the rest of the world, will be able to overcome this really horrible pandemic. And thanks for listening. I'll be happy to take questions. Okay, and so I think I'll read the questions from the Q&A session. Okay, so please do elaborate. Why was there such a difference in the spread of SARS coronavirus 2 and MERS or SARS? So that's an excellent question, and I'll tell you what we think is reason for that today. So the one reason is that SARS and MERS are more lethal than SARS coronavirus 2. Okay, so MERS and SARS both have a mortality rate of about 10% or higher, which means that the virus actually is less able to spread. So if a virus kills its host, then usually it can't continue, right, to spread. And so unfortunately, SARS coronavirus 2 is in the sweet spot where it transmits very, very effectively, but it actually has a relatively low mortality rate. We're talking about 1% or even 5%, which are the highest estimates of mortality rate. That's lower than MERS and SARS coronavirus 1. So that's reason number one, why those viruses spread less. So they simply kill their host. The second reason is that SARS and MERS spread most when patients are symptomatic. And that makes life easier because when you see someone who's symptomatic, you can quarantine them, right? And if you see someone who has a fever, you can tell them you can't enter whatever gathering now. Whereas this current pandemic, SARS coronavirus 2, the virus transmits at a pre-symptomatic phase. It's already very, very transmissible before symptoms occur. And so that makes it very, very hard to control spread because, someone feels that he or she are perfectly fine. They're actually pre-symptomatic and they spread the virus. And so those are the two reasons why it's thought that this virus could not be controlled and those other two viruses could. What makes a virus more transmissible and less pathogenic? Again, an excellent question. The short answer is that we have no idea. But just to emphasize, the mutation that I mentioned, B614G, it doesn't make the virus less pathogenic. It's as pathogenic as the G form. So we have the D form and the G form. One with a D, that's an amino acid, right? An amino acid at position 614. Another with a G at position 614. They're both equally pathogenic, but the G form apparently is able to. It's a kind of complex biology of which I can't get into the details, but the G form makes more virus. And so if it makes more virus, the virus is transmitted more efficiently. And that's currently what we know about specifically about that form of virus. How should different COVID therapeutic strategies, vaccine, viral entry blockers, viral proteases consider the mutations of SARS coronavirus 2? Again, excellent questions. And so there's a huge focus on tracking the different mutations in this pandemic. The mutation that I just mentioned, the D614G, it was detected because there's a specific dedicated effort to look across all the mutations that have been found so far and to try and correlate them with vaccine design, with entry blocking, with viral proteases. Okay, so there are dedicated efforts only for that. Currently, there has been no evidence whatsoever that any of the mutations that have been observed so far have any effect on vaccine design or on developing viral entry blockers, except that one mutation that I mentioned, the D614G. The D614G does not have an effect on the vaccine design because it doesn't affect the epitope, any of the epitopes that the vaccines are targeting. It affects a different region. And so that's kind of my answer. Do you have any suspicion on who can be a super spreader? Could it be more related to behavior than physiology? Yeah, excellent question, and that's what we're following up on now. So my research now is trying to understand what makes a person a super spreader. I definitely suspect it's also behavior and also physiology. Okay, so I think it's a combination of both. We think it also has to do with viral load. So it's literally the number of viruses that a person bears. Okay, so someone who has a very high viral load, and it's not only the viral load, but it's where the viruses are located. And so viruses that are in the lungs are actually probably less transmissible. Okay, someone has to really cough very, very efficiently, let's call it, or very, very violently to create to allow these viruses to exit this person. Whereas people that have a very high viral load in the upper respiratory tract or in the upper parts of the nose, they might be super spreaders because it's easier for them to simply emit virus simply by talking or breathing or singing, which is it turns out a very, very efficient way of spreading virus. And we have other kind of lines of study that we're now following. Unfortunately, I have no answers. Do you think the tracking system has been sufficiently elaborate to estimate the transmission dynamics? Is there anything you think it should be improved in terms of testing, social rules, etc.? I think there are differences in different countries across the world on tracking systems and how they estimate transmission dynamics. I think the more you test, the better you can track the virus. I think it's the most important rule, just to test as much as possible. Now, clearly testing is not enough because you have to test and then you have to cut the transmission chain, right, if you want to stop the transmission dynamics. I don't think this is exactly what was asked, but I'm following up on this. So the most important thing is to break these transmission chains, right? We have to detect virus and then stop the transmission of the virus. And unfortunately, as I mentioned, because the virus is pre-symptomatic, that creates a huge challenge because most people are tested when they feel sick or when they develop symptoms. And so we have to test even more rapidly and broadly than that. I think that the social rules of masking, of course, are extremely, extremely important. Masks work. They simply work. The more people are masked, the safer we are, and the more social gatherings we prevent, the more people we can save. Why do transmission dynamics vary so much between different countries, but with the same social distancing rules? Is it because of the genetic variables of previously applied policies on vaccinations or the social behavior? I think it's a mix of everything. So what I can say, again, specifically when seeing what goes on in Israel, I am confident that social behavior has a huge effect. So I can tell you what's going on in Israel. Okay, so unfortunately in Israel, there's a lot of lack of compliance. A lot of people disobey the rules. There are large gatherings here that are performed because a lot of people perceive this illness as an illness that will affect only elderly people. And so young people have parties and they think that they're going to be fine, which again might be true. Statistically, young people are less affected, but there's no way of preventing, right? So if you have a party and you have someone that is sick and that person afterwards goes and meets his or her parents, and his or her parents are older, and they will meet the neighbor, and there's no way of stopping this, okay? And so eventually the only way is really to perform the social distancing. And so I think social behavior is playing a huge role here, and that's explaining differences among different countries. And I think that there is definitely a role also for genetics, okay? And I think it's starting to emerge that the background genetics of the infected people is also affecting the disease, but it's very, very early, okay? And so I'm saying this based on my intuition more than on scientific facts. Okay, what do you think about doing massive tests at asymptomatic persons? Again, I think it's important to massively test asymptomatics, but it's as always a measure. It's economic decision, right? How many tests are possible in a certain country? I don't think I have anything additional to say other than that. Doesn't mean that the more intense one's infection is, less is the threat they cause to others in terms of transmission and vice versa. Not necessarily, okay? So you can also you can develop very severe disease where you have virus in the lungs, but you also have virus in the upper respiratory tract, okay? And so unfortunately, it's a combination of both of these. Did you apply a similar sequencing study in a cohort of patients for the second wave in Israel? No, we have not applied a similar sequencing study in the second wave. We've started to, but we've sequenced a much smaller cohort. But we don't have anything to say. All I can say is that from what we can see very clearly from the second wave is that the second wave is a direct continuation of the first wave. Okay, and so because the borders are still closed in Israel, or mostly closed, what we see in Israel is we can see that it's the same transmission chains that began in the first wave. They're simply continuing to the second wave. Due to the sequence similarity of the SARS-CoV-2 and other coronaviruses, is it possible that the people who are covered for other virus infections before might have a better immune response against SARS coronavirus? Again, an excellent question. So this has been studied. We know that the antibody response against SARS coronavirus 1 does not neutralize SARS coronavirus 2. Okay, so that's antibody response, or what's called humoral response. But the other response that is under intense investigation now is what's called the T-cell response, cellular response. So we know that the other arm of the immune response are cytotoxic T cells that are able to detect virus, and there is accumulating evidence that this arm of the immune response, there is cross-protection. So there might be people that were infected from the common cold in the past, might be also protected from the current SARS coronavirus 2. Unfortunately, this entire field of immunology is much less developed than antibody responses, and so we don't really know the extent. Okay, so these are experiments that were performed in tissue culture. So in tissue culture, we can see a very strong response of T cells against both the coronavirus from common cold and SARS coronavirus 2. It is not clear how this affects real live people, okay? We know that a lot of people were clearly infected by the common cold, but we don't know how many of them really are protected. It's one of the theories why children might be more protected. So children develop a much less severe form of disease, and one of the theories is that maybe they have a strong T-cell response because they have the common cold very regularly, but it's only a theory right now. Cool. I managed to answer.
Yeah. Thank you, Adi, for the amazing talk, and thank you all the audience for posting so many interesting questions. And if we don't have any question anymore, then we will have a lunch break, and after that, we will have a talk from Sammy Jeffrey. And see you later. Thank you.
Great. Thank you all. Bye.
Bye-bye.